Hello students, welcome to this channel and I am Anuradha Kashyap, your biology mentor. I hope you all are doing very well. Today in this video, I am going to share a topic which is very important with respect to your NEET and also with respect to another exam, like if you are preparing for CSIR NET, if you are preparing for CUT, then this topic becomes a very great topic. Why? Because it is a topic which is related to your 12th standard, which is related with the topic that is biotechnology. And in the biotechnology unit, there are a lot of techniques which we will are going to deal one by one. So in this series of technique of biotechnology, there is one topic, and that topic is your agarose gel electrophoresis. This is a very great chapter to study. And in this topic, in this video, I'm going to tell you about the topic, about the concept behind the topic, what is the principle of the topic, and what is the basic structure of that instrument, due to which we are going to perform that very kind of technique. We are going to perform a technique which is related with your biotechnology. Okay. So before starting the video, I just want to tell you the concept behind this topic. And before starting this concept, I am talking about DNA. If you know very well that DNA is a kind of biomolecule which is basically carry negative charges. Okay, DNA is a double stranded helical structure which carry negative charge due to phosphoric ion, due to phosphate ion. You all know this very well. Okay, and when we talk about DNA, so the DNA is basically a large biomolecule. But when we want a gene, which is a desired gene, then the size of the desired gene is small. Normally, the size of the desired gene is small with respect to your big DNA. So, if there is a big DNA, if there is a big DNA, and we want our desired gene, which is small in size, then we have to find out that fragment out of those big ocean of DNA molecule, right? So in biotechnology, there is a tool that is restriction endonuclease. Don't worry, I will tell you everything about agarose gel electrophoresis by showing my presentation, all the pictures, all the images which I taken here, all are taken from all are taken from Google search. Normally, all are the Google images, and all the references goes to Google. Okay, Google images, Google sites. So before starting, I am just want to make a interaction between us so that you can know very well about this concept, right? Okay, so let's start. First of all, I want to tell you that there is a concept, there is a technique that is Agarose gel electrophoresis, and before Agarose gel electrophoresis, you have to know about DNA. DNA is a big molecule. DNA is a big biomacromolecule. That's why we are calling it macromolecule, because its size is very large. Okay, but when we talk about our desired gene, that it is always a small in size. So first of all, from the large fragment of DNA, you have to cut your small desired gene, and by using restriction endonucleases, which is a tool in biotechnology, you can cut your desired gene from the big fragment of DNA. Okay, so first of all, you are using restriction endonucleases, but restriction endonucleases make lot of cuts there, right? And when you are performing any kind of experiment, then you are not taking only one molecule of DNA, you are taking lot of molecules of DNA, right? So when you are taking lot of molecules of DNA and you are using restriction endonucleases, then lot of cuts are also obtained, lot of fragments are also obtained, and these fragments are very different sizes. You will obtain lot of different sizes of DNA fragments. So when you find out your small desired segment, then which technique you will use? So this is the technique. This is the technique. Basically, students, this is the technique that is agarose gel electrophoresis is those technique which is used to separate out that small segment of DNA from the pool of your large segment of DNA. Okay, so this is the basically technique, and there is a principle there is a concept behind your agarose gel electrophoresis. When I'm saying that I am going to separate out, I am going to separate out my desired gene, which is a small molecule, from the large fragment, from the large pool of the DNA, then what will be the principle behind it? Actually, this technique follow a principle, that is the size of the DNA fragment, or we can say the size, the DNA fragments are can be separated. The DNA fragment can be separated on the basis of their sizes. DNA fragments can be separated on the basis of their sizes. Small molecule DNA fragment will be obtained at the end of the instrument, at the end of the media, and large fragment of DNA will obtain near the starting sample of the DNA. Okay, so I hope some students will be confused at that time. At this time, students are confusing that what is a sample, what is near, what is away from the sample. Okay, so just give me 1 second. I am telling you. Okay, so student, first of all, we are going to separate what we are going to define the term, that is agarose gel electrophoresis. I am going to tell you all these things in a very simple way. Okay, so this is our concept, electrophoresis. This is the basic name, basic definition. We can't say definition, it is the name of the technique, agarose gel electrophoresis. So first of all, what is agarose? What is agarose? So students, agarose act here, agarose in this technique, agarose act as a media. What is media? When you are doing any kind of experiment, you are forming, you always produce, or you always form a basic media. Okay, supportive media. Supportive media, you can say, it can be made up of any kind of gel, or it act as a nutritive media for some kind of bacteria. For example, when you are making a culture plate, okay, when you are making a culture plate in a petri dish, you are making a culture of any kind of bacteria, then you are using a kind of media which is very nutritive. This media contains a little different kind of amino acids, sugar, and salt, and lot of vitamins, lot of vitamins, peptones, yeast extract. All these things are present on those kind of media. Okay, so that your bacteria can feed upon those nutritive media, and can grow and can make lot of colonies. Right, that is your culture media. But when we talk about here, we are talking about a supportive media. What is your supportive media? Here you are using a kind of agarose. What is agarose? Agarose is a jelly-like substance, or you can't say it jelly-like substance actually. Agarose is a kind of material which is obtained from plant. Plant. I'm talking about plant, but it is not a plant. It is a kind of algae. From algae, we are going to obtain this material. We are going to obtain this semi-solid, semi-liquid material, which is your agarose, and it acts as a nutritive media. Okay, so first of all, this is your media. Which kind of media it is? It is your supportive media. Supportive media. And student, when I'm talking about a supportive media, then it is your agar, agar. Sometimes we are saying it polyacrylamide gel, we can say it polyacrylamide gel. Okay, but here it is your agar. Agar is obtained from, writing it here. Agar is, all these questions are already asked in it. So agarose is basically agar, and it is obtained from a seaweed. Sorry, it is obtained from a red algae. Red algae. Okay. And Agarose is also called as agar agar, and we can also say it seaweed. Seaweed. Great. So agarose is a media which act as a supportive media, and we can also call it agar agar or seaweed, and it is obtained from a red algae. So ma'am, which is the example of red algae from which from where we can obtain it? The example you have to remember, red algae are Gracilaria. I hope it is visible. Okay, I am going to write it here. Example of red algae from which agar agar is obtained. That is your Gelidium and Gracilaria. Good. So these two terms you have to remember. Now, next, what is the gel? You know very well when you are applying gel on your hairs. Then what is the consistency of that media? Basically, it is not as much solid and it is not as much liquid. So it is semi-solid or semi-liquid in nature. We can say it either semi-solid or semi-liquid. So basically, what is a gel? Gel is a media, or gel is a compound. Gel is not as much solid and not as much liquid. So it is a consistent, like honey. Honey. Gel is basically what it is, semi-solid, and you can say it's semi liquid kind of thing, semi solid or semi liquid kind of thing, right? Next is your electrophoresis. When we break this word electrophoresis, okay, let me choose this color. Electro, when you talk about electro, so basically what is electro is your electricity or power, or you are going to apply electric field. In physics you already read about electric field. When you are going to apply electric field, you are creating a potential difference, and you know very well that when there is a potential difference, then charges start to move toward different opposite voltages, okay, different cathode, right? So phoresis, what is the meaning of phoresis here? Okay, so what is the meaning of phoresis? Phoresis is basically Phoresis is basically movement. Students, what is the meaning of phoresis? Phoresis is basically movement. Movement of molecule, or which kind of molecule? It is your charged molecule. Charged molecule. And you know that I am talking about DNA, and DNA is basically, it is your negatively charged. So, okay, I will write down all the properties of DNA in respective slide. Okay. So this is the meaning of your technique, that is agarose gel electrophoresis. And when we talk about this technique, we are going in detail. Technique which is used in biotechnology. A. Tiselius was the discoverer of agarose gel electrophoresis. He was the first person who discovered about agarose gel electrophoresis. Okay. So one question will comes in your mind then, that what will be the instrument, which kind of instrument we are going to use to perform this kind of a technique, right? So this is the diagram of the instrument. Here you can see this is the diagram of the instrument. Okay, first of all, I'm going to hmm, student, I am going to tell you about the whole process of agarose gel electrophoresis, and then I will cover all these things in one flow. Okay. So first of all, basically what happens here. So when I'm talking about agarose gel electro, electrophoresis, right? So here we are here a kind of vessel. You know, you have all of you know about shoes box, right? You are image you image sorry you imagine that you have a shoes box, right? And this is a kind of box. This is not right. Hmm. Done. Give me 1 second, student. Actually, pen tab is not working. Now it's good, right? So this is what? This is a kind of box. It is a kind of box, a vessel. You can say it a vessel. And in this vessel, there are two kind of electrodes are present. One is your positive electrode and one is your negative electrode. Please tell me positive, what we will say positive electrode. Positive electrode. This is a positive. Suppose it is a positive electrode and suppose this is a negative electrode. Negative electrode. So you know that positive electrode is called as anode, right? Positive electrode is called as anode. This is an instrument and this instrument have two electrode, two ends, and on the ends two kind of electrodes are attached. One is anode and one is cathode. Cathode is negatively charged electrode and anode is a positively charged electrode. In this electrode, we are going to add our supportive media, which is your what? This is your agarose, or we can say it agar. So first of all, you are taking you are taking. I'm going to write it here. Okay, you are taking 2% agar and agar powder. Basically, you are using agar powder, which is obtained from a algae, that is red algae, that is Giladium or Gracilaria, and we are also calling it as seaweed. Now 2% agar powder add in test tube. You are adding some buffer inside it and then you are boiling it. When you are boiling it, then it will make then it will start to develop consistency, right? And it develops into a semi solid or semi liquid form, right? Now this semi-liquid form from the from the test tube, you are adding inside this vessel. You are adding this inside this vessel. So we are saying it that a boiled agar is we are going to add here, right? Now, as it is a boiled agar, so it will occupy the space inside the box. So complete box is filled with agar, right? Complete box filled with agar. Before cooling down this agar, when you are adding this agar inside this box, at that moment you are adding a kind of comb. What is a comb? In your house you are using comb, right? So this kind of comb you are adding at one end of this vessel. You are adding that on. You are adding that on towards cathode. You are adding this towards cathode. Suppose this comb you are adding here. Suppose this comb you are going to add here, right? Now, when this agar becomes solid, when this agar cool down and becomes solid, at that moment you are removing that comb from there. You are removing that comb from there. Then what will left behind? In the agar, some sorry, some holes will develop. Some holes will develop there. Some holes will develop here. For example, you are adding comb here, and when you are removing this comb, then there then at that place agar will not fill, and then you will obtain some kind of holes here, right? You will obtain, you will find out holes here. At which end? You will find it at cathode. Now these holes are obtained. Now these holes are obtained. Now you are going to add your sample. What is your sample? Your sample is a fragmented DNA molecule where lot of different kind of fragments are present. Some fragments are very big and some fragments are small. And you know very well that that small fragment is your desired gene and you have to separate it, right? You want to separate it. That's why you are adding your sample, which you don't know that which is where is your small sized DNA fragment. So you are adding a mixture of your sample inside these holes. Inside these holes, you are going to add your sample in very small amount, that is in nanogram amount. So you are adding your sample here. Suppose you are adding your sample here. Suppose you are adding your sample here, which is what is your sample? Different kind of DNA molecule. It can be the large or it can be the small, right? So this DNA sample, I am writing it here, DNA sample in ng nanogram, okay? And if you want to track the movement of your DNA sample, how will you know about the movement of the DNA sample? Actually, this complete process, this complete instrument is now in resting stage. But when you switch on, then electric field will be provided towards cathode to anode, and at this condition, your DNA segment will start to move over the agarose gel, over the agarose gel, right? So DNA sample in nanogram amount you are going to add in your holes, which act as a sample. But when you want to track the movement of the DNA, you have to add some kind of dye so that you can basically know about the movement of your DNA, right? So as you are adding sample in nanogram, you are adding some kind of dye also here, and this dye is your you are using basically two kind of dye you are going to use. You can use two kind of dye. One can be your okay, so one can be your ethylene blue, ethylene blue, and second kind of dye can be etherium etherium bromide. This is very important. Etherium bromide. This dye is highly important because it is asked in it every time. Mostly time this question is asked about etherium bromide. So etherium bromide is a kind of dye. I'm writing it here. It is a kind of dye which will gives you orange band, right? Suppose you know very well that your DNA is double stranded. Your DNA is, I am making a rough diagram. Your DNA is double stranded, and in between both strand of DNA, hydrogen bonds are present so that one nucleotide can attach with another nucleotide and can make a complementary basis sequences, right? So there is a hydrogen bond in between. So how this DNA will obtain a kind of color, how this DNA will appear as a orange when we are using this dye. So basically this dye intercalated in between the hydrogen bonds of this DNA. So this dye intercalate between this dye intercalate, suppose this is the dye. Now this dye intercalate between hydrogen bonds of DNA. I am saying that what? DNA, Ethereum bromide, Ethereum bromide dye intercalate at hydrogen bonds of DNA, which will give them orange color. But students, this orange color will not, this orange color will not cannot be seen in visible light, cannot be seen in visible light, cannot be seen in visible. I hope you are making notes. Cannot be seen in visible light. What is the visible light? Between 400 to 700 nanometer range. Light is known as wavelength. Light is known as your visible light. So how can we see? How can we see these orange bands? This Ethereum bromide dye intercalate hydrogen bonds of DNA, and these orange bands can be seen. Orange bands can be seen in UV light. So this is a very great concept. You have to remember that now this instrument is in resting stage. But when I'm going to switch on, I will switch on. Then at that moment, this DNA sample starts to move over the agarose gel electrophoresis. Now, how can they move? How can they move? So basically, agarose gel shows a sieving effect. So, ma'am, what is the sieving effect? So here you can see sieving effect are basically like a network. Suppose this is a kind of network, and these are the pores. These are the pores where DNA molecules will embedded inside it. DNA molecules when starts to move, okay, when DNA molecule starts to move from negative to positive end, because in a DNA sample, DNA is basically DNA is negatively charged ion, and it is hydrophilic molecule, so it will attract toward positive end, attract toward positive end, right? Attract toward positive end. So at cathode, where cations are present, at cathode you can say anions are present, and at anode cations are present. Cations are basically positive charge, and anions are what? Anions are negatively charged, right? So this DNA sample in nanogram, when you are adding here, and you will add ethylene blue, and you can add ethylene blue or you can add Ethereum bromide. Ethylene blue or you can add methylene blue. Two kind of dyes are present. So what is the function of ethylene blue? You already know about the function of ethylene bromide. I told you that ethylene bromide is a kind of dye. When it interact with DNA, then it intercalated between the hydrogen bonds of the double stranded DNA, and it will appear as a orange color, not inside the visible light. It appears as orange color inside the UV range, UV light, right? But what is the function of methylene blue? Actually, what happens? All In every school, in every institution, it is not possible to make a UV chamber. So basically, what they will do, they perform this experiment with the help of ethylene or methylene blue dye. Actually, this dye is known as a tracking dye. You This is a tracking dye. Actually, it is a kind of tracking dye. It is a kind of tracking dye. So as your DNA will start to move over the agarose gel electrophoresis, this dye will start as a blue. This dye will appear as a blue color, right? So your DNA bands, the movement of DNA band can be checked out with the help of this ethylene blue. So it is a very easy way with respect to your Ethereum bromide. You can avoid Ethereum bromide as a dye. You can use methylene blue as alternative option, right? So by using this tracking dye, you can track the movement of your DNA sample. So when you start electric field, when you apply electric field here, suppose I'm going to apply electric field here. Suppose electric field is applied here. So there is a major voltage differences occur, right? A voltage difference is created and your DNA sample will start to move. Now DNA sample is negatively charged and start to move toward anode that is positively charged, which is situated far away from your negative ends, right? So what happens as it starts to move, then agarose shows a sieving effect. What is the sieving effect? This network-like structure. Agarose is a kind of material which always shows a sieving effect. It is very important question that agarose shows a sieving effect. That is, it creates pores inside it. And when DNA start to move over these pores, then big fragments of DNA will embedded, will entrap inside these pores. And the molecule which is small in size, the DNA molecule which is very small in the size, it will start to move fastly over the agarose gel electrophoresis, right? So I'm repeating it all over the situation in a once file, in a once flow. Suppose, okay, students. So saying, I am telling everything in one flow. There is an instrument, and this instrument is known as electrophoretic chamber. In this instrument, two kind of electrodes are situated. Two kinds of electrodes are present. One is cathode, one is anode. Cathode is negatively charged, anode is positively charged. Cathode is negatively charged due to presence of anions, and anode is positively charged due to presence of cations. Okay, so you are adding agar here. You are going to add agar here, which is in boiled condition, and agar is obtained from a red algae, that is Gillidium and Gracilaria, which is also called as a seaweed, right? Now, when you are going to add a boiled agar here, and you are waiting for you are waiting till their cool down. When it starts to cool down, before cooling, you are adding a kind of material which can make pores inside the agar. Now, at one end, this comb will form some pores, and in this pore, now you are going to add your DNA sample, which is already cut by restriction endonucleases. So different segments you have. Some are big, some are big, some are small. Your desired gene is small, so you want to find out your small desired gene, right? So you are adding your sample. So to track the movement of DNA over the agarose gel electrophoresis, you can add two kind of dye. One will be your methylene blue, which give blue color to the bands, or you can say your moving DNA. It will give blue color, and you can add your Ethereum bromide. Ethereum bromide is a kind of dye which produces orange color bands under the UV light, right? Why? Because it intercalate with the hydrogen bonds of the DNA. That's why it produces your orange color inside the UV light. So when I'm going to applying electric field, so voltage differences is created, and due to this voltage differences, the DNA start to move over the agarose gel electrophoresis. Inside the as it is present in 2% condition, that's why agar will form some pores inside it, and it will produce a sieving effect. Due to this sieving effect, large DNA segment remain, or you can say entrapped inside the pores which is near to the cathode, and the small segments of the DNA will move over those large fragments and reach towards the anode. So the smaller the size of the DNA, it will move faster over the agarose gel electrophoresis, and the larger the segment of the DNA, it will move very slow over the agarose gel, right? So the large segment of the DNA will obtain near the cathode, and the small segment of the DNA will obtain near the anode. This concept is very much clear to you, that large segment of the DNA will obtained at the negative end of the electrode, and small segment of the DNA will obtained at the negative end of the anode. Sorry, a small segment of the DNA will obtain at the positive end of the electrode, right? Of the chamber. Now, How can you measure? How can you compare your bands with another bands? So basically, in the in different lanes. Suppose this is a standard lane. I am talking about a standard lane, and this is lane number one. This is lane number two, three, and four, and five. Right? This is five. This is five. So this is a standard lane, and standard lane, over the standard lane bands are already appear. Suppose these are the bands. Okay. So it act as a standard lane. It act as a standard lane. And when you are going to add your sample inside these wells, so you will obtain different kind of, suppose a different kind of band. Suppose this for one lane you are obtaining band here. For second lane you are obtaining band here. For third lane you are obtaining bands here. Bands of what? Bands of your DNA molecule. And for the fifth, for fourth lane, for fourth lane you are obtaining your segment of DNA, bands of DNA here, which appears orange in color. And finally, for fifth lane, you are obtaining your segments here, segment of DNA here. So by looking over this instrument, you can see, you can find out that your desired gene is basically what is your desired gene is the, your desired gene is this. Why? Because it is small in size. That's why it is showing fast movement over the agar, and it reaches toward positive and of anode, right? So this is the desired gene, and when you are separating your desired gene, this is again very important. When you are separating your desired gene, or you can say desired fragment of DNA, plus means you are obtaining your desired gene with some gel, that is agarose gel, then this process is called as elution. This process is called as elution. So I hope this is very pretty much clear to you that what happening here. Now you can see about the images. You can see over the images. Okay. So this is first image. Here you can see this is electrophoretic chamber. This is electro. This is electrophoretic chamber. With this chamber, you can see two electrodes are attached. One is anode and one is cathode. Cathode is negatively charged, anode is positively charged. It attracts DNA because it is positively charged, that's why it attracts DNA. This is your polyacrylamide, or you can say it agarose gel, and you are adding buffer solution here also. So when you are going to apply electric field, you are going to give it a power source, then from the sample DNA starts to move towards anode. It will start to migrate from the negative end towards the positive end according to their size. So when anyone asks to you what is the principle behind agarose gel electrophoresis, then you have to answer that fragments of DNA, fragments of DNA are separated, fragments of DNA are separated according to their sizes, right? Answer according to their sizes. And small segment of DNA or small DNA toward which electrode? Toward anode, which is basically positive charge, and large DNA, large DNA will obtain toward toward cathode, toward, right, which is your negative end. So this is the real chamber. You can see here. This is electrophoretic chamber and this is power source. Now you are adding buffer here, and this is basically the vessel where you are adding your supportive media, that is your agarose gel electrophoresis. This is a diagrammatic representation. This is solution. You are adding gel. You are this white color representing agarose gel, and these are the sample wells where you are adding your sample, which is mixed with your methylene blue, or you can use your ethylene bromide, and these are the electrodes, and this is the movement of the DNA towards arrow representing the movement of the DNA from the negative end towards the positive end. And finally you are obtaining you are getting a kind of you are getting a kind of chart on which it is not actually a chart, it is a supportive media where you are getting bands. Is your adenine, this lane representing cytosine, this is lane representing guanine, and this is for thymine, this is for adenine. Longer bands are present near the wells and shorter bands are present away from the wells, right? So here you can see this is the negative electrode, this is positive electrode, and you are adding this, you are applying electric field which create a potential difference 60 to 120 volt, and these are the DNA samples which are representing by blue color here, and these are the standard lane. This is your standard lane. By using the standard lane you can compare all these kind of bands, right? And separation, for the separation, for the movement of the DNA, you have to run this electrophoretic chamber for 30 to 45 minutes actually. You have to wait for 1 hour actually, so that you can obtain this kind of result, right? Now this is your agarose gel and you will obtain this kind of result here. These are the size marker. These size marker, you can say it's standard lane, you can say it's size marker. And these are the different position where bands are obtained. As the molecular sizes of the DNA is very high, very if the molecular sizes of the DNA is more, then it will cover less distance. It will cover just this distance. But if the molecular sizes of the DNA is low, then it will cover these distance. It means shorter DNA fragment can move farther away from the sample well, but the segment of the DNA which are larger in size, they cannot move as much farther from the sample well. They will present near the sample well, right? And again, this is a kind of representation where you are adding mixture of DNA molecule. These are called as append drops, and you are using pipette. By using pipette, you are going to add these DNA samples into the sample well. Mixture of DNA molecule of different sizes you are adding here, and you are also adding some methylene blue or dye here. You are applying the electric field power source, and this is the gel, and you will obtain this kind of result. Shorter molecule will obtain at the end of the gel, and larger molecule will obtain near the sample well. This kind of instrument, that is your pipette, different kind of measuring pipettes are present inside a lab. So you are using pipette by adding a nanogram amount of your DNA sample. You are adding this sample into the sample well, or you can say into the holes or pores which you have already created when you were um, by adding foam, right? By adding foam. This is the whole procedure of the agarose gel electrophoresis. First of all, this is a casting tray which is also called as glass plate, and over the glass plate you are adding agarose solution, that is you are going to make a masking tape, and after forming a masking tape, this you want to pull it down, then it will form solidified agarose gel, and comb is pushed. This is the comb is pushed down into the gel to form wells. Now wells are formed, and you are adding your sample with methylene blue or with your ethylene bromide, which will give you orange color. Now DNA segment loaded into the wells with micropipette, and you will obtain this kind of result. Different kind of bends are obtained here. Okay. So I am very fast right now because I already explained everything by writing here, right? So this is a much faster view, and you can look over this actual result. This is a standard lane, and under the UV range, UV light, you will see these orange color bands. When lot of molecules of same size present at the same well, then the intensity of this will become very high. So here you can see some are bright, some bands are very bright, and some bands are not as much bright, right? So these are sample wells, and you already added sample here. Now after applying electric field, this sample starts to move, and and after 1 hour you remove this gel from the casting tray, from the electrophoretic chamber, and you will took this agarose gel, solidified agarose gel, inside the UV rays, under the UV rays, and then look over them, right? So this result will appear there. Now you can see this is the standard lane. These are the sample well where you are testing first, second, third, fourth, fifth well, and the molecular sizes, the DNA which have molecular sizes 6,000 base pair, 5,000, 4,000, 3, sorry, 600, 500, 400, 300, 200, 150. So you can see 50 base pairs. So here the desired gene will be this or this, because their molecular sizes is less than 400 base pair, right? So you can find out this technique very easy. I hope this is very clear to you right now. And one thing is that when you remove these, when you remove this desired gene from the sample, from the gel, or you can say when you are removing this result, when you are removing your desired gene with gel, then this condition is called as, this process is called as elution, which is again very important, right? So this was all about your agarose electrophoresis. I hope this is clear. Now if you want to find out if you want to find chapter of NEET, regarding NEET, right? So you can follow my another channel, that is Easy NEET Crack. Easy NEET Crack is my another channel. You can follow me there. If you only want to prepare for your NEET 2024, then you can follow that channel also. And this is my basic channel. Here I am going to upload all kind of videos which will be beneficial for every student who is preparing CSIR, who is preparing for their NEET, who is preparing for their CUT, who is preparing for any kind of science subject, right? And who just want to increase their knowledge. So these are the material which I am going to provide here. So if you like me, if you like my content, then please like and subscribe. Thank you, students.